My first research was actually based here at Newdegate Research Station, where we're located today. When I first came to Newdegate, it was a highly important research centre with a lot of micronutrient work, so I was very lucky to start off my career here. So one of the major experiments that we had at the Newdegate Research Station was called 66N14. Started many years before I actually started in the department, but it was very important because it was an experiment where we were able to measure how long copper lasted in the soil. Up until that time, farmers used to apply the micronutrients, mainly copper, every 5 years. So it was an experiment where we were able to actually extend the length of time that copper lasted in the soil and remained adequate for both crop, pasture and animal nutrition. So it was a very important trial, and I was lucky enough to start off my career at that point in time, where I was able to sample and get the results and analyse the data from that particular trial. It was also a point where the micronutrients were being plant tested, and the youngest fully emerged leaf of a wheat was being used to diagnose copper deficiency. It's probably one of the biggest breakthroughs that I've seen in my career in the department. And I was lucky enough to work with people from University of Western Australia, Alan Robson and Kevin Snowball and Jack Lonergan, who developed the test, and I was able to partake or be in part of that particular research as well. So it actually helped me develop my career in the area of plant testing. I was then offered a position at Esperance, which then still allowed me to come to Newdegate Research Station because it's about halfway between Perth and Esperance where I was based. So my work here at Newdegate still continued into the mid-90s, so it was certainly a very valuable part of my career to look at the residual value of the trace elements for cropping in WA. The research that happened here at Newdegate Research Station on the particular trial at looking at the residual value of copper certainly lengthened the time where farmers had to apply or reapply copper. So instead of putting it on every 5 years, they were able to go out to 15 or 20 years. In the mid-90s, the trial still continued, and I recropped the area in the early 2000s, and we were able to clearly show, because of the experimental area allowed us to do it, that we were able to measure the copper was still effective. The applications that we put on, the recommended applications that the farmers put on in the mid-60s were still adequate for cropping in the mid-90s. So we were looking at a residual value of something like 30 years. So instead of applying copper, or the need to apply copper every 5 years, we then extended it out to 30 years. So it certainly changed the amounts and the rates that copper were applying, farmers were applying at that particular point in time. Something else that I thought about, and which was very interesting in my research work, was the effect of the size of the granule which contained the copper and the distribution through the soil. The farmers in this particular area had often what they call new land copper problems, and we found that it was actually the size of the granular fertiliser that they were using and its distribution through the soil. A whole lot of research that was done in this Newdegate area, we clearly defined that how the copper was distributed and the size of the granular dramatically affected the amount of copper that the plant was able to access. So large granules often ended up with copper deficient, although recommended applications of copper had been applied to the soil. So we recommended at that particular time that the trace element mixes that were made in the state contained certain quantities of fine material so the plant could access copper distributed through the soil. So fairly conclusive results and sort of factors which did affect the new land development through this particular area.